Thank you very much, Ms. Anja Rath. Thank you very much, Mr. Somchai. Welcome to all our guests today. Thank you very much for joining in our joint webinar. This is a joint webinar from the company KK Scientific in Thailand and the company Memmert. My name is Andrea. I'm joining the company Memmert for more than 15 years, and I will be the speaker today in regards to our topic, stability tests according to the ICH guideline Q1A. Today, we will cover information on stability testing, stability zones, stress and storage conditions, and important factors. And these important factors, this will cover also climate chambers, which can be used for the stability testing. So now we will go ahead with a presentation. And after the presentation, there will be a live demonstration on our product, on our latest product, HPP Echo. Now, I would like to invite you to come along with us and to join our webinar today. Thank you again for being with us. Let's start with the presentation right now. Stability testing, stability zones, stress and storage conditions, important factors. When we talk about the stability testing according to the guideline ICH Q1A, then this covers the new drug substances and the products, the finished products. There's several chapters giving information on the stability testing. And when we look closer to the ICH guideline Q1A, we will find some linkages which are referring to the ICH Q1C. Here you can find information for new dosage forms. There is ICH Q5C. This refers to biotechnical and biological products. We have ICH Q6A and ICH Q6B. There's included a list of tests and references to analytical procedures and proposed acceptance criteria. And there's ICH Q3A. There's included a list for the degradation products. Furthermore, when we talk about stability testing, there comes up some details in regard to the quality guideline, which means also quality guideline Q1B. In Q1B, we can find information on the photostability testing of new drug substances and products. When we talk about photostability testing, we are talking about the testing along with light. We have Q1D. Here, there will be given information on bracketing and matrixing. These are useful concepts and are important processes in minimizing the quantities of samples of drug formulations and products assessed for stability. Then there is Q1E. That means here we can find evaluation of stability data. That means there will be given recommendations how to use stability data generated according to ICH Q1A, and it contains also examples of statistical approaches to stability data analysis. Q1F, this is now also very interesting. In this Q1F, this is a guideline, and this guideline actually have been withdrawn. The intention of this was actually that there was the wish to harmonize intermediate storage condition in climate zone 1 and 2 with long-term condition in climate zones 3 and 4. And there was no consensus because there have been some countries, they said, they want to have more strict conditions because here was based on the testing conditions of 30 degrees and 65% RH. But there have been some countries, and their countries said, we want to do more strictly testing. We want to do testing under higher humidity range. And that came up that now we have actually 5 climate zones because we have climate zone 4A and 4B. And the climate zone 4B, this refers now to the very strict condition of 30 degrees and 75% RH for long-term testing. And this refers climate zone 4B also to the Southeast Asian countries. So that means also for long-term testing, Thailand is doing the testing based on this condition of 30 degrees and 75%, what the long-term tests concerns. However, this has been withdrawn, but this condition has been kept for the intermediate testing. So in the ICH guideline Q1A, there's intermediate testing included. And here, these conditions, 30 degrees, 65% RH, has been used for the intermediate testing. Yeah, then there's 8 memoir. That is in regard to qualification and validation. Then we also have CFR, Title 21, Part 211, 137 for the expiration dating. And we have Title 21, Part 11, 211166 for stability testing, and 211170 for reserve samples. That is in regard to the ICH guidelines, where you can find stability information on stability testing. Of course, in your country, you have also the Asian guideline, and you are following the details as mentioned in the Asian guidelines. Let's go further to the ICH Q1A. Here we have International Council of Harmonization of Technical Requirements for Pharmaceuticals for Human Use. The ICH is an organization, and this organization has by now 17 members and 32 observers. It wants to bring together the regulatory authorities and pharmaceutical industry to discuss about scientific and technical aspects of pharmaceuticals. Also, it would like to develop ICH guidelines and achieve greater harmonization worldwide to ensure that safe, effective, and high-quality medicines are developed and registered and maintained in the most resource-efficient manner whilst meeting high standard. And of course, when we talk about the ICH guideline Q1A, it refers to the package what is requested for the registration application. So we have here stability data package for the new drug substances and products to be defined, and which is sufficient for a registration application within the 3 regions of Europe, Japan, and United States. And that means when we talk about this ICH Q1A, this refers to the climate zone 1 and 2. So ICH Q1A always covers climate zone 1 and 2. Let's go further. Let's have a look on the drug development process. When we talk about drug development process, then we have here several steps. We start with the discovery and preclinical development. Afterwards, they come and goes on the clinical trials. And of course, after that, we have the post-marketing testing. We have then the product, which is NDA approved. And then there will be on the ongoing stability tests. When we talk about the preclinical development, that means a lead compound has been identified. So here, the stage of research that begins before the clinical trials, which is the testing in the humans. And here you have to consider that actually there is a lot of compounds and only one enters, for example, this drug discovery stage. So it could be more than 5,000 compounds until there is recovered a compound which can be used for the further steps. So the stage of preclinical development, which finally becomes an approved drug, this is called IND. And this IND means investigational new drug. That's from the United States Food and Drug Administration investigational new drug program. And the pharmaceutical company obtains then the permission to start with the human clinical trials and to start the heavily regulated process of clinical testing in human subjects. Then we have here the clinical research phase. The clinical research stage, which is representing the time from the beginning of human trials to the new drug application. And new drug application, that means that is the abbreviation NDA. And this can actually take more than 10 years. So it's a long period of time until a new drug is available. When we consider the clinical trials, that is a type of research that studies a test or a treatment given to people. The clinical trials studied how safe and helpful tests and treatments are. And when it's found to be safe and helpful, they may become tomorrow's standards of care. And we have here these different phases because we start first with a small group of volunteers, then there comes more groups and more people. So phase 2 means 100 to 500 volunteers. Phase 3, we have here 1,000 to 5,000 volunteers until we get then the approval for the new product. And the most important step during this drug development process is the pharmaceutical analysis and stability studies. So that means stability studies is already done in the very early stage and is a very important matter when a new drug and a new product is manufactured. Of course, when we talk about stability testing, we have to consider this always refers to documentation. So documentation is a very, very important matter also when we talk about stability testing. Yeah, what will be done when we get a new drug? How is this development process? When we talk about this development process, we have an API that is the active product ingredient. And everything what is not the active product ingredient, that is an excipient. And that could be substances formulated alongside the active ingredients, and it could be bulking agents or fillers that could be lactose, paraffin, water, ethanol, polysorbate, cellulose ethers, which is, for example, used as thickener or soft capsules or talc. And then the new drug substance, which is active, that will be tested for stability and impurity. And then there comes the formulation development process. And this is the composition of the drug product that contains then the API and the excipients. And this drug formulation, this could be then available in various forms, like, for example, in a solid form or in a semi-solid, in liquid. As we know, we have capsules, we have tablets, we have emulsions, we have suspensions or oral powders or inhaled intranasal. So that is in regard to the drug formulation. And then afterwards, the finished product, of course, will have to be checked in regard to stability. So here we do the formulation interaction, the excipient compatibility, and further we have the next step, which refers to the packaging. And here the packaging also is a very important factor, because the packaging can have an influence on the quality of the product. And of course, when we talk about stability testing, the most important is always that we get a very safe product which can be used, because we talk about the safety of the patients. So that means here we have a primary packaging that can be ampoules, vials, containers, blister packaging, plastic or metal, which is in direct contact with the product. Then we have the secondary packaging. That is the packaging which is used as an additional layer of packaging, and that is normally requested for hygiene purposes, for preservation or for protection of the product, and it will not be in direct contact with the product. So that refers to the packaging, which also has an influence on the product. So always determine and assure the identity, potency, and purity of ingredients, as well as those as formulated products. Let's go on. Stability testing. Stability testing means discover how a drug product or as a drug substance changes over a defined period of time under certain conditions as temperature, air, humidity, and light. Because these conditions, they can have an influence on the quality of the product, on the safety of the product. So that means establish retest period for drug substances as well as the shelf life, and recommend storage condition for drug product. So that means now we have to find out how long can the product be used, how long is it saved, and which are the storage conditions for the product. And it is a regulatory document which is according to ICH, FDA, and of course GMP has to be followed during the full process of manufacturing. Ensuring pharmaceutical product is safe and effective, irrespective of where in the world it will be supplied. And this, I think we all know, you can get APIs from different countries. You can manufacture your product. You will export it to the different products. So we follow guidelines and rules to have the safe products and which are going to be tested according to the conditions in which the product are then also finally going to be used. In ICH Q1A, the information stated thus: provide evidence on how the quality of a drug substance or a drug product varies with time under the influence of a variety of environmental factors such as temperature, humidity, and light, and establish a retest period for the drug substance as well as the shelf life for the drug product and recommended storage condition. And here you also see there's a division in regard to drug substance and also in the finished product, the drug product. So let's have a short summarization. And the summarization means what means stability testing and shelf life? This question we can find on our next file. So here, what does it mean? This is a method for checking the quality of the product and to analyze how long it retains its quality. It is used to determine if the quality of the drug substance and drug product is altered over time under the influence of the environmental factors. For drug substances, the stability testing determines a retest period, which is the time frame during which the drug substance is expected to remain within the specification, and therefore it can be used in manufacturing of the drug products. And for drug products, the finished products, stability testing is necessary to fix the shelf life of the product, which of course also means here to consider also the labeling, the correct labeling, how long it can be stored under which conditions, and how long it can be used to be safe and retain the therapeutic value. Let's go further. Let's go to the next chapter and let's look a little bit closer in the ICH guideline. One chapter is in regard to the stress testing. Stress testing, another wording is also forced degradation studies. And the forced degradation is an exposure of the drug substance or a drug product to different stress condition which results in relevant degradation products. So here we are considering the degradation of the product itself, and that means we can help to likely get identification of the degradation, and this is useful to establish the degradation pathways and the intrinsic stability of the molecule. So here it is very important that we know about the behavior of the molecules, that we understand the chemistry of the molecules, to do the investigation, stability related property of an API, and develop and understand the degradation products and pathways. That is important because based on this information, we can go on with our studying on the product itself. And then we also will know exactly how is it going to be changed, what we have to consider. And furthermore, it is also an integral part of information provided to the regulatory authorities in registration application dossiers. So stress testing is important, is a part for the ICH guideline, and it's important to know about the molecules and to find out the method, because also in the beginning we don't have a stability indication method. So here is also necessary in order to get and to gain a SIM stability indication method. Let's go further. This is not mentioned in the ICH guideline, but this is giving even more information. What is all included when we talk about these degradation studies? It means elucidates the structure of the degradation products. So that means to find out about the degradation products, develop and validate a stability indication analytical method, identify impurities which are related to drug substances or excipients, generate more stable formulations, distinguish degradation products in formulations that are related to drug substances from those that are related to non-drug substances, solve stability-related problems, generate a degradation profile that mimics what would be observed in a formula stability study, facilitate improvements in the manufacturing process and formulations in parallel with accelerated pharmaceutical studies, choose a correct storage condition, appropriate packaging, and better understanding of the potential liability of the drug molecules. So there's quite a lot of details mentioned for the stress testing. And in the ICH guideline, there is mentioned, like we see on the next file, so here is mentioned, it should examine the effects of temperature in 10 degrees increments, e.g. 50 degrees, 60, that means above that of the accelerating test. The humidity to be examined should be 75% RH or greater. It should be carried out on a single batch of the drug substance here. Note, we are talking about single batch. Testing in solution should also be performed across a wide pH range, either as a solution or suspension. These samples are then used to develop a stability indication system. And photostability testing should be an integral part of stress testing. And in general, it means testing under more severe conditions than those used for accelerated testing. This comes up to my next question. And the next question means now, should photostability testing be an integral part of stress testing? When we think about stress testing, now we say here, should it be an integral part of stress testing? And yes, it should be an integral part of the stress testing. And when we talk about this photostability testing, then we refer to the ICH guideline Q1B. In the ICH guideline Q1B, there is mentioned about the exposure of the product, and the exposure, this should be done in regard to the cool white fluorescence and near ultraviolet lamp. And for this procedure, there is going to be used a product for which you can do also photostability testing. And Memmert has here, for example, also a product that is called ICHL. And here there could be done photostability testing. In this guideline, there's mention about the exposure in regard to the cool white fluorescence lamp, which should have unspecified and similar output like ISO 10977 and a near UV fluorescent lamp, which has a spectral distribution from 320 nm to 400 nm with a maximum energy emission between 350 and 370 nm. So that is mentioned in this ICH Q1B, and that refers then also to the information about the energy, which means here we are talking about an overall illumination of not less than 1.2 million lux hours, and for the ultraviolet energy of not less than 200 watt hours, QM hours. So that is mentioned in Q1B, this information in regard to the photostability testing, and more details we can also give on another seminar, which is more linked then also to ICH Q1B. Let's go further. Let's have a look again on the force degradation study. Let's look to the drug substance and to the drug product. Drug substance, which could be solid or it could be solution suspension. When it's solid, then there will be done tests in regard to photolytic, thermal, which refers to heat, thermal and humidity. If it comes up to the solution or suspension, then we have here exit and base and hydrolysis and oxidative. Oxidative degradation, that means we use hydrogen peroxide. That could be in the concentration range of 3 to 30%. When we talk about hydrolysis, then this refers to the chemical degradation reaction of the analyte with water. Then we go further to the drug product. Drug product, we have here a solid, semi-solid solution suspension. Solid, there will be considered photolytic, thermal, and humidity oxidative. Semi-solid, here is considered photolytic, thermal, and humidity. And solution suspension, photolytic, thermal, and oxidative. Important when we talk about this degradation study is to identify the primary degradation and not to completely degrade the API. So that means you should generate some of this degradation, but not completely. And that really have to be considered. So that means a condition for this degradation should be typically about 10 to 30% loss of API as determined by assay when comparing with non-degraded API. In the total absence of degradation products after 10 days, the API is considered stable and particular stress conditions. So that was in chapter in regard to the forced degradation study. Now let's go further. Now we come to the next chapter in the ICH guideline Q1A, and that refers to the storage testing. And that means when the objective of the stability study is now defined and the method for the capability is now certain, the drug will be evaluated on a much larger scale. And now we start with stability study. And we talk about this stability studying. Then it comes to long-term studying and accelerated studying and intermediate studying. Long-term, that is a real-time studying. That means that the drug substance or product is stored at recommended storage conditions. And there will be done monitoring until the specification fails. And accelerated testing, the study is designed to increase the rate of chemical degradation or physical change of a drug substance or drug product by using exaggerated storage conditions. The data from accelerated and long-term studies, they can be used to assess longer-term chemical effects at non-accelerated conditions and to evaluate the effect of short-term excursions outside the label storage condition that it might occur during shipping. Because what we have to consider, if we manufacture our drug and until it's going to be used at the, let's say, customer or the patient, then there's a long period of time. There could be transport included. You could send the product to a different country. During the transport, we have different climate zones to pass. And that means that also we have to be considered until there is really lifetime. So that means how long it can be used and how long it is safe, even during these different conditions. And we have intermediate testing, this intermediate testing, which is 30-65%, which comes into effect in case the accelerated test fails and the long-term testing is done in under 25 degrees and 60% RH. Let's go to the next file. Here we can see information on all the different climate zones available on the globe, and also some information and details under which conditions the testing will be done. So here we have information from WHO and ICH Q1A. We refer to the long-term testing. If we go climate zone 1, it says 21 degrees, 45% RH. If we say 1 and 2, it is 25 and 60% RH. If it's climate zone 3, the long-term testing will be done at 30-35 degrees. If we talk about climate zone 4A, it is 30 degrees and 65% RH. And climate zone 4B, here the testing will be done at 30 degrees and 75% RH. To consider the worst case means that there could be covered climate zone 1 and 2, and that's what also in the ICH guideline can be found. So in order to cover climate zone 1 and 2, we come up to 25 degrees, 60% RH. In order to go for intermediate testing, we have 30 and 65% RH. And in order for climate zone 4B, which of course is not mentioned in ICH guideline, this is separately at 30-75% RH. Let's go to the next file. Let's have a look to the different climate zones. Climate zone 1, that is temperature climate, which refers to Northern Europe. Climate zone 2, that is subtropical and Mediterranean climate, like for example, Southern Europe. Climate zone 3, this refers to hot and dry climate like it might be in Iraq. Climate zone 4, which is hot and humid. Then we have climate zone 4A, which is hot and humid climate, which is, for example, Yemen, or 4B, which is hot and very humid climate, that is Brazil, or for example, also Southeast Asia. So let's go on to ICH Q1A, because as mentioned, ICH Q1A, that refers to climate zone 1 and 2. And in this guideline, there is mentioned long-term testing will be done at 25 degrees, 60%, or 30 degrees and 65%. It's requested to do the testing for 12 months until a registration process can start. And important is there is a remarks mentioned, which means you must stay within a certain temperature and humidity range. When it goes to the temperature, we must stay in a range of ±2 degrees. When it comes up to humidity, we have these limits of ±5% RH. Then also we have accelerated test, which is done at 40 degrees, 75% RH for 6 months. And also here we have some limits, ±2 degrees, ±5% RH. And intermediate testing, that comes into effect in case the accelerated storage fails. Then there will be done intermediate testing, also based on the fact that it is done on long-term 25-60. So if we have long-term 25-60, accelerated 40-75, we have an error, then we go ahead and we do intermediate testing at 30 degrees and 65% RH. Yeah, let's go further. We have here different chapters. So that means storage in refrigerator. So if the goods are for storage in refrigerator, then the long-term conditions for testing will be 5 degrees in a limit of ±3. And the accelerated testing will be done at 25 degrees, 60% RH. And here we have the limits of ±2 degrees and humidity ±5% RH. This storage in refrigerator, that could be products which are intended for this refrigerated storage, and that could be, for example, vaccines, which might be stored under refrigerated conditions, like, for example, DNA oligonucleotides. So that is, for example, an example for this product which is stored in refrigerator, for which you have to do the testing in long term at 5 degrees. Let's go further. There's another chapter. This refers to the storage in a freezer. That is actually very rare. But also here we're talking about the DNA oligonucleotides. Also here there could be done testing, and that means when it comes up to this products, then conditions will be at -20 degrees, marks ±5% limit. And furthermore, there's mention about drug substance or drug product for storage below 20 degrees. And this is also very, very seldom. That's why we also have a case by case. Nowadays, it comes, for example, in case when we talk about the BioNTech Pfizer drugs, for example, which are going to be used, because here we have conditions of -75%, -75 degrees. Yeah, then further, we have to consider what kind of packaging we have. It is impermeable or is it semi-impermeable? Considering on the sensitive to moisture or potential for solvent loss, that is here's a concern. And if the product is packed in impermeable containers, the testing can be done under more controlled ambient conditions. So here, like mentioned, impermeable container, sensitive to moisture or a potential for solvent loss is not a concern. It can be conducted under any controlled or ambient humidity condition. If we go further and if we have a look to the semi-permeable packaging, this allows some material to pass through, but this allows other materials to pass. So there could be aqueous-based products which are packed in these semi-permeable containers, which should be additionally to physical, chemical, biological, and microbiological stability evaluated for potential water loss, also for the safety of the product. And this means here we have then the testing to be done long-term, 25 degrees or 30% RH, 30 degrees, 35% RH. Accelerated test is here at 40 degrees, 25% RH. And if it comes to intermediate testing, we have here 30 degrees and 65% RH. Also here we have the remarks and limits. So for long term, it is ±2 degrees, humidity ±5% RH. Accelerated is in the limit of ±2. And if it comes to intermediate, then also here the limits of ±2 and ±5% RH are requested. So evaluate for potential water loss in addition to physical, chemical, biological, and microbiological stability evaluated under low humidity. Let's go on to the next one. Now we come again to the next question. And the question says, is it compulsory to do both long-term and accelerated testing for the drug products? Yes, and this is correct. It is always compulsory to do both. So you need to do the long-term testing and also accelerated testing in order to do then issue also your registration package. So you need to do both. That is necessary and requested according to the rules and guidelines. Let's have now a look to the next file. Here we can find an example in regard to a stability report. In this stability report, it comprises the primary stability data of 3 registration batches. So here we are always talking about 3 registration batches for a product. This is packed in PVC, PVDC blisters. And here we have ICH guidelines considered. That means testing is done at 25 degrees, 60% RH for 12 months and 6 months at 40 degrees, 75% RH. By this means climate zone 1 and 2 is considered. And here we have a result which says analytical procedure was stability indicating and completely validated. No change in appearance, disintegration time, dissolution range. The average mass increased of absorption of water 1.2%. The hardness decreased slightly. The active ingredient degraded slightly, 0.4%. After 6 months at 40 degrees, 75% RH, 0.15% after 12 months at 25 degrees, 60% RH. The degradation product was formed. Its structure is elucidated. It is qualified up to 10%. It has no influence on the safety. The container closure system was faultless. So that is an example for this stability report. And that gives here an information. Furthermore, we have here also an example in regard to the test protocol. When we look up this test protocol, you can find that there's mentioned the 3 different batches for the registration. Also the storage conditions is mentioned, the storage period in regard to testing frequency, and also there would be some testing specification. That is the example on this testing. Now let's go on to the next file, which says also ongoing stability study is a part, because after we have done the registration process, of course, we must go on with the studying. So we cannot just say, now we register our new API, now we register our new product. After that, of course, you still will have to go on with studying. And that means now we go on with ongoing stability studying, which is to monitor the API and to determine that the API remains and can be expected to remain within specifications under the storage conditions as indicated on the label within the retest period of shelf life in all future batches. Here there's also an example in regard to the labeling. So here labeling says do not store above 25 degrees. And when we consider the storage labeling and the testing condition, then of course this must be identical. So here the testing has been done at 25 degrees, 60% RH for the long-term testing and accelerated 40-75% RH. And that means here recommended labeling, do not store above 25 degrees. Yeah, that was in regard to the guideline. Now let's have a look further. When we consider the guideline, then we also have to consider how can we successfully do our stability testing? What is necessary for successful stability testing? Of course, the personnel and staff is important. It is a careful checking of all the data is very, very necessary to follow GMP guideline, but also, of course, the climate chamber you are using for stability testing is very, very important for successful stability testing. And this climate chamber should, of course, meet all the rules and guidelines, and it must be a safe product. And now let's go further and let's have a look to the next files, which are referring to climate chambers. Here, I would like to introduce you our new constant climate chambers, HPP Eco. This is a product which is based on Peltier technology. And this special technology, that means we have a maximum energy efficiency compared to climate chambers with a compressor. It is very important to have absolutely homogeneity distribution inside in regard to temperature and humidity. It must be reliable and fail-safe because we're talking about this testing, which takes really long period of time. So of course, it is important that you have a safe product. And always a good climate, refrigerant-free, smooth-running, compact, low maintenance. So to perform stability study successful, you have to have a good quality stability chamber meeting all the guidelines and rules. And our HPP Echo, which is from our latest development, this was introduced by this year in the market with an advanced Peltier technology, because Memmert is actually using Peltier technology already for a long period of time, for more than 20 years. So we have a lot of experience with the use of this technology. So now we further have done developments on these products. Let's go on with the next file to have a look on this technology and to see also even more these outstanding features of this product. Here we can see a chart which gives an information on the energy saving with the new advanced Peltier technology. And you can see here on this chart different kind of bars. The gray bars refers to the energy consumption in regard to a product based on compressor device. The red bar here gives an information to Peltier devices in regard to energy consumption, and the Peltier device mentioned here, which refers to our HPP 410 Echo, that refers to the green line. And when you look to this sketch, you can see, for example, if we use here temperature 25-60% RH, the gray bar comes up to almost 6,000 kilowatt hours per year. If it goes to the Peltier, then we might come to around 1,000 kilowatt hours per year. And if we look for the green line, then you see it's very, very low. So that means here the energy consumption is really a very important aspect, what we considered at Memmert, because for us, the environmental aspect is very important. So to look for future technologies, it is very important also to have products which meet the future technologies, and energy consumption is certainly one of the factors which will have to be considered also for the future. Let's go on to the next file and let's have a closer look in regard to additional improvements, what we have done on our HPP Echo, ecological and economical product, because when we talk about this Echo, one aspect is environmental aspect. Further aspect is if we have low energy consumption, also the costs for operation is lower. So that is also be considered here. So we have this new Peltier technology, which is highly efficient. Furthermore, we considered a new humidification system. This humidification system refers now to even more precise control of humidity. So that is very important for us to have a mostly precise humidity control, and also to reach the humidity in a very fast way after opening the door. And there's a new airflow routing system that refers to the homogeneity inside, because that is also very, very important when you do stability testing. When we talk about this humidity inside, it means we have, for example, we measure at the different points inside to see what is the temperature deviation. And here we consider a new system also in order to get a very good result. So that means outstanding homogeneity due to changed airflow routing in the interior, faster recovery times after opening the door, optimal temperature distribution, even at maximum load. And let's go to the next one. Let's have a look which products we have. We have HPP starting from size 110 up to HPP 2200. This means we have small constant climate chambers, and also very huge one. This very huge one, which has approximately 2,140 liter. So that means this is a really big chamber. And even for this huge chamber, we use the new Peltier technology, the advanced Peltier technology for cooling and for heating up, because that is always one principle for which you can go up and cool down. We have also, of course, considered on our products the documentation, which is very important. And that means all our products, they have storage. The storage means up to 10 years every minute. There will be done the storage of the set data and also the actual data. There comes a software along with the product that is called Atomo Control software. And if you have to follow the FDA confirming regulations, then we have also a software confirming to FDA 21 CFR 11. Yeah, that was in regard to these constant climate chambers. Let's have a look here on the pictures again. So you can see starting from size 1110 up to the size 2200, which is a really very tall product, which comes then also with these heated up glass doors. We have these chambers, which are going to be used for stability testing. Important, of course, when we talk about stability testing and the features of the product, to consider the temperature humidity diagram for which the products can be used. These products are mostly useful for the stability testing in regard to the guidelines and also for these long-term testings in the different climate zones. Now, this was a presentation on the product. Let's now go on with the live demonstration, because we have here an HPP Echo, and I would like to show and give you more details in regard to our HPP Echo product. HPP Echo. We can set here the temperature range by touching the area. We just turn the knob and we confirm it. So it's very intuitive and very easy for controlling. When we talk about climate chambers, the second parameter, which is very important, is humidity. So just touching the area, turning around the knob, and you can set the humidity. You can see here the external canister. This will be connected, and then you have the humidity control in the unit. We furthermore have here information on the alarm. So that means we have an alarm for temperature and humidity, and we can set a high alarm and a low alarm for the humidity as well as for temperature, and also an ASF. So that means multiple protection for the product. If we go under menu, then we have here the information in regard to the correct time zone. This is now important when we do the documentation. So always make sure that the correct time and data are mentioned. And then furthermore, under the setup, you can see IP address. This means all of our products have an IP address. When you set up the LAN connection with your computer to read out the data, so you have here IP address, which is here important. Yeah. In regards to our product as well, let's have a look to the interior. Of course, made out of stainless steel, like all of our Memmert products. In the interior, we can see advanced Peltier technology at the rear. Next to the advanced Peltier technology, you can see further Peltiers, which are used for the dehumidification process, because also dehumidification is important, which also has been improved on this product. Then on the sides, you can see temperature sensor and over temperature protection sensor. We have two sensors. These are two PT100 sensors for wire measurement in Class A. These two sensors communicate with each other, and they can even take over the operation. And we have a humidity sensor, which is a capacitive sensor. Then very briefly, I also would like to show you a little bit how it looks like at the rear, because then we can see an information on the new pump. One moment. Okay, let's look. Here we can see again the Peltier technology. Here we have a tray if you collect the condensed water, which can be taken out. Peltier for cooling and heating, and Peltier for dehumidification. And we have here the micrometering pump. Micrometering pump, which is also a new feature, which refers to the humidification of the unit, and which means mostly precise humidification and most fast humidification. That is here also considered on our constant climate chamber. And as you can see, this constant climate chamber does not have any compressor. So this constant climate chamber is based on this Peltier technology, which is our new advanced technology, which is used by Memmert. Our new model advanced eco Peltier technology. So this was all in regard to our seminar today. Now let me go on and let me also ask Mr. Somchai if there are some more questions from your side. And let's have now a part in regard to the questions. Hello, Somchai.
Some question. Somebody asked, is stress testing and accelerate testing are the same?
Stress testing and accelerated testing, right?
Yeah.
No, this is not the same.
Testing are the same?
Okay, thank you. No, it is not the same. So we have differences. Stress testing and accelerated testing is differently. The stress testing is done under even more severe conditions in accordance to accelerated testing. Accelerated testing is done at 3 batches, and the stress testing is done on 1 batch. And that means in order to find out the method, in order to find the stability indication method, then there will be done this stress testing, which is also called forced degradation studies. So this is even under more severe conditions in comparison with the accelerated testing. So it's not the same, it's differently.
I see. The next question. At which temperature range has long-term testing can be done in Thailand?
Okay, very good question. In Thailand, it refers to the climate zone 4B. In Thailand, we have the Asian guideline to follow. And here, the long-term testing for the generic case is done at 30 degrees and 75% RH.
Yeah. The next question. Can SPP model, SPP eco model, can be used for stability test and photo stability test?
Okay. So the HPP Echo is for stability testing. It can fulfill the rules and regulations to be used for stability testing. If we talk about photo stability testing, which means testing with light, the HPP Echo cannot be used. Because HPP Echo, we cannot do with UV light and daylight. We have a possibility to integrate an LED light in HPP Echo, but not UV light. If UV light is requested and to check on the, let's say, conditions like outside, Then we need UV light and also daylight. There we have another product, and this product is called Climate Chamber ICH. This is our product also for photostability testing.
Oh, see, that.
Mm-hmm.
Okay, thank you.
Yes,
And
you're welcome.
the last question is where we can find technical data on climate chamber of Memmert.
Okay. Yes. Mm-hmm. Very good question. Yes. I think technical data is really one very important point you must consider. So these technical data are available from our side, and also KK Scientific. They have all the technical data available. So please kindly contact Mr. Somchai. He will be very glad to give all the technical data requested. And we have really extensive technical data on all the different temperature points and humidity points. So please kindly ask Mr. Somchai. He will be very glad also to forward technical data. Mm-hmm. Yeah. Maybe some more questions, or if there's not more questions from your side, then I would like to say thank you again. Thank you, Mr. Somchai Konat, also for all your help and support in regard to this webinar. We hope that we could share and give some information. So we will be also glad to do future webinars also as well. And then I would like to thank very much KK Scientific team and all participants for being with us today. Yes.
On behalf of the audience, we also would like to say thank you to you for all your effort to give us knowledge today. I hope you will come to give us more knowledge again in the future. Thank you so much.
You're welcome. Thank you so much. Thanks a lot, and see you next time. Okay.
Bye.
Bye. Bye. Bye from Germany. Bye.